Thank you so much, and thank you to everybody here tonight, this afternoon, for staying such a long time in this meeting. It's been an exciting time, and I've really enjoyed the papers, and I want to particularly thank the organizers for inviting me. I've been to several of these previously, but never as a speaker. I'd like to talk to you today about the genomics and engineering of non-conventional yeasts for biotechnological applications. I'm going to break that down a little bit for you in the first few slides. But first, I'd like to introduce Tom Kelleher, who is our CEO at Xylome, and my wife, Giovanna, who actually helps out along with our four stalwart employees at Xylome. And I'd also like to thank Robert Riley and Sajid Hardras, and particularly Igor Grigoriev. Igor, because we've participated in several different projects now, and particularly also to Meredith Blackwell and Cleet Kurtzman, who initiated the main study, along with myself, the main study on biotechnologically significant yeasts. How do I advance this with the green button? In any event, I'd like to also say, yeah, is the green button? Okay. I'd also like to just say that Cleet Kurtzman unfortunately passed away just this last year, and left a big hole in the area of yeast taxonomy. So with that, biotechnological applications start with problems or opportunities and markets. Without markets, you don't have biotechnology. Now, just to point this out, in rough numbers, the beer fermentation in the United States is about $108 billion a year in sales. Now, that's all attributable basically to one organism, Saccharomyces cerevisiae or Carlsbergensis. They're close enough that you could consider them more or less the same. Wine, likewise, $60 billion a year in sale in the US. And this compares to industrial ethanol at about $22 billion. Now, that assumes that we're making about 15 or 16 billion gallons of ethanol per year. Now, that's pure ethanol, and it's being sold at about $1.35 a gallon. It does not include any of the animal feed byproducts that go into it. If you were to make cellulosic ethanol, the potential industry for cellulosic ethanol, just in terms of the potential feedstocks that are there, would be about 5 times that, or about $100 billion a year, maybe a little bit more. Industrial enzymes, by comparison, and this includes all the enzymes that are made for starch saccharification, along with the enzymes for basically detergents, which is the major, and any other enzymes which you guys buy, I buy on a daily basis, about $8 billion. So you can see the biotechnology and fermentation industry is large, but it really doesn't compare to pharmaceuticals. The total pharmaceuticals industry in the United States is about $333 billion a year, with patented sales of about $244 billion. These are 2016 numbers. And biologics, which are the recombinant biochemicals that are used, everything from human growth hormone to epogen, neupogen, and all of the other peptides that make our health and chase away cancer these days, are somewhere between $80 and $110 billion a year. The difference being that those materials sell for about $1 a microgram, as opposed to $1.35 a gallon. So the biotechnology industry really depends on getting from lower value products into higher value products. Now, ethanol production from cellulosics has problems. There are problems. There are also opportunities, but there are problems. People might be surprised to think that or to hear me say that there's not very much biomass available. And by that I mean not the total amount of biomass that's out there, but the biomass that's consolidated and concentrated in regions of the country where you can economically convert it into a sustainable industry, where you could collect it, transport it. It takes about all of the corn stover from a 30-mile radius around a commercial-sized cellulosic ethanol plant in Iowa, where the corn densities are the highest. Not only that, but when you pre-treat this material with either acid or whatever you're using, autohydrolysis, it makes toxins, the most conspicuous of which is acetic acid, but also various other things, ferulic acid and various degradation products from the soluble lignans. Enzymatic hydrolysis is expensive and slow. And actually, in all of the years of trying to discover new cellulases, I don't think very many people, if anyone, has actually improved on the turnover number of cellulases to any significant extent. That's because the cellulose kinetics or cellulase kinetics are largely determined by the substrate and not by the enzyme. As a consequence of the low bulk density, sugar concentrations are low. And even if you engineer yeast to use xylose and all the sugars that are in there, they're still sensitive to acetic acid, which, as I said earlier, is made directly from the hemicellulose in the feedstock. Now, industrial ethanol production from grain presents an opportunity because grain is cracked, steamed, and enzymatically digested, then fermented and distilled into ethanol. And the resulting mash is centrifuged to produce two things, basically: distillers' dried grain feed and stillage with soluble organics. And these materials, this simply indicates that the corn is taken to the plant, and all of those organics end up at the plant. And there's no additional transport that's necessary. Not only that, but you've got all the heat, and waste heat. You have all the licensing, the air and the water that's there. So what you're seeing in the cellulosic ethanol field right now is what they call Gen 1.5, which is halfway between using cellulosics and using fibers from starch. Industrial ethanol plants generate about 30 billion kilograms of organic byproducts annually, and they're mostly located in the upper Midwest. Thin stillage is the major one that we're focused on. The syrup is sprayed onto grain residuals or spread onto soil, and its value at 4 cents a kilogram or 2 cents a pound is dropping. This is because disposal is a major problem. There are low margins and limited market. The industry knows that it must diversify. Now, Xylome's yeast process solves these issues. Our organism makes enzymes that break down the stillage organics, and it converts them into lipids. It helps recycle the water, which is actually a very important element at these plants, because even though you're making, the stillage is coming out of those distillation towers at a rate of 400 gallons per minute, 60 minutes an hour, 24 hours a day, 350 days a year. It's a huge water flow, and even in the Midwest, we can't sustain to just throw that material away, and the high concentration of organics makes it a disposal problem. However, we can take those materials and turn them into a fish oil or a palm oil substitute. So we're taking these low-value organics from this thin stillage and getting a 16 to 60x increase in value to make high-value products as a palm oil substitute or as an omega-3 fish feed. Now, Xylome is developing technology based on non-conventional yeasts. Why palm oil substitute? Well, the palm oil market is $65 billion a year. And there's a growing demand for substitutes because, frankly, palm plantations are not sustainable. They all occur in the tropical regions, and they're destroying lives, they're destroying wildlife, and they're destroying whole communities. The aquaculture market is it's expected to be $200 billion by the year 2020. It's fast approaching that. but it requires fish meal to supply omega-3 fatty acids. So we've specifically targeted this market. Now, yeasts are among the most important microbes in biotechnology. I've already pointed out that Saccharomyces cerevisiae makes as much in value as just about everything else put together. And Pichia pastoris is used for heterologous proteins. Candida utilis is a fodder yeast. Yersinia lipolytica, which has been mentioned before, is used in cheese making. Clavieromyces lactis, Deberrymyces hansenii are both used in cheese making. Jeffries, also known as Pichia stipidus, is a pentose fermenter. So there are a lot of different yeasts that have different characteristics that can be used in biotechnological applications. The objectives of my talk here are to put these into a phylogenetic context and relate the physiological properties to the genomic features. If we look back at traditional strain development, starting at about the 1950s to 1975, the way strain development occurred was to discover traits in native isolates and amplify those through mutation and selection. That was it. Just taking one organism and pushing it by random genetics and selection until you got what you wanted. Metabolic engineering really took off in the 1980s to 2000, where we were introducing exogenous genes into organisms with known genomes and physiologies. And for the most part, we were confined to the use of model organisms, such as E. coli and Saccharomyces cerevisiae and Pichia pastoris and a few other things like this, where we were making specific proteins, individual proteins, but then that gradually moved into introducing whole pathways. Probably one of the first attempts at metabolic engineering was to put xylose isomerase into Saccharomyces cerevisiae in order to make it a pentose fermenting yeast. That actually did not succeed for approximately 20 years for reasons that we did not know until it was very recently shown that a chaperonin was necessary in order to stabilize the xylose isomerase in the heterologous Saccharomyces cerevisiae background, but that's a whole other story. Our contemporary practice really kind of started in 2000, where we have extensive use of genomic information to expand the natural capacities of non-conventional organisms. And in this way, we start off with organisms that actually pretty much do what we want them to do, and we try to make it better. But what this means is that we have to develop whole new genetic systems, transformation toolboxes, do the metabolic engineering and modeling, and then we have to work in a different physiological background. Now, why is this important? Let me give you just a simple example. Genetic features can fundamentally change the physiology of the cell. In the case of respiration in most fungi, you have the NADH dehydrogenase complex, or complex 1, which takes NADH and it uses a proton pump to generate a proton gradient, which then comes back across the membrane and generates ATP. And the presence of the NADH dehydrogenase complex enables production of more ATP for protein. In Saccharomyces cerevisiae, however, complex 1 does not exist. It's gone, which means two things. First of all, it means there's a lot more NADH in there to use for alcohol production, and it also means that there's less ATP for protein synthesis. This is why Saccharomyces cerevisiae is actually not a very good organism in order to produce proteins. That's why Pichia pastoris is used so much better. Loss of complex 1 is associated with fermentation, along with the substitution of urine, the loss of urine 9 and the acquisition of uro 1 from lactobacillus, which enables Saccharomyces, along with several other mutations, to grow anaerobically. And Saccharomyces and a few, very, very few other organisms are exquisitely, almost uniquely among eukaryotic organisms capable of anaerobic growth, although it's still limited. Now, our study, in which most of my talk is based, looked at, I believe it was about, we were looking actually at about 18 organisms. The ones that have significant biological potential are shown here in bold. Our study went down to Lipomyces starkii, which turned out to be the basal yeast in this particular case. Lipomyces starkii is actually more closely related to Aspergillus than it is to Saccharomyces cerevisiae. And it also included the CTG clade of yeasts. And in the process, although I'm not going to describe it, we also discovered a new codon system. So the CTG yeast, of course, substitutes serine for leucine. And we found another one, the Pachysolon tenaphilus, that also has a different codon system. But I don't have time to describe that. If you would like to know more about that, look for Comparative Genomics of Biotechnologically Important Yeasts, our paper that was published in 2016 in PNAS. Now, the pentose phosphate pathway, when somebody is talking about metabolic engineering of a pathway, it's important to understand that you're not just looking at one or two genes. In this particular case, going from xylose down to ethanol is 18 metabolic steps. And you can't just change one or two of those steps and expect the physiology of the cells to change. For example, you have this whole pentose phosphate pathway up here, which is supplying NADPH to drive this assimilation reaction, and it's having to compete with the Embden-Meyerhof pathway. Now, Saccharomyces simply goes straight down the Embden-Meyerhof pathway. 80% of the carbon goes in this route, probably 80% plus. Relatively little goes through the pentose phosphate pathway, but in most fungi and yeasts, in fact, the pentose phosphate pathway is predominant. So what makes pentose fermenting yeasts different? There are two pentose fermenting yeasts that I've spent some time studying. One is Spathospora pachylidarum, and the other is Jeffrysomyces stipidus. And of these two, even though they're very, very closely related taxonomically, of these two, Spathospora pachylidarum is clearly the better xylose fermenter. And it lies, at least in part, due to these first two enzymes. Now, the first enzyme in this pathway, xylose reductase, would use NADPH as the cofactor for reductant. In both of these organisms, either of those enzymes, not the xylitol dehydrogenase, but either of those xylose reductases can use either NADH or NADPH. So the question is, really, what is the affinity, the relative affinity of those enzymes in metabolism? And here you see, this was a paper that was published by a young woman, I don't know her first name. Hu was her last name. She published as a sole author in Applied Microbiology and Biotechnology. I really know not much more about the origins of this paper, except that she has some really very interesting data. She compared the xylose reductase activities, and this was in crude homogenates, of Spathospora pachylidarum to those of Pichia stipidus. And if you look first at the xylose reductase, you see that the affinity, the Km, for NADH is 17.3 micromolar, whereas the affinity for NADPH is 31.7. Now, if you remember your biochemistry, the lower the number, the higher the affinity. So this shows you that there's twice the affinity for NADH over NADPH on xylose reductase in Spathospora. If you look at the same relationship for xylose reductase in Pichia stipidus, you see that it's 20.9 for NADH and 18, I'm having difficulty reading it even from here, 13.3 for NADPH, meaning a much higher affinity for NADPH than for NADH in Pichia stipidus. And if you look at the affinity for xylose with NADH, it's 17.1, whereas with Spathospora and NADPH for Spathospora on xylose is 52.2, which means that with NADH, it has a higher affinity for xylose, and you compare that to the same thing in the case of the Jeffrysomyces stipidus, and you find that those ratios are just completely inverted. Going then to the xylitol dehydrogenase, you see that, again, with the xylitol dehydrogenase, and this enzyme it's actually a secondary alcohol dehydrogenase. It doesn't have an NADPH activity. But you see there, the NAD affinity is 39.2, as compared to the NAD affinity or Km of Pichia of 86.8. So clearly, just changing those affinity ratios, not talking about changing the activities, changing the Km of the enzyme completely transforms the way these cells handle xylose, because Spathospora pachylaterum actually is shoving the xylose right on down the system, and Pichia stipidus is piling it up as xylitol. And this is something I give you this example because a lot of times what we do when we're doing metabolic engineering is we go in and we kind of say, okay, a little bit of this enzyme is good, but what's better is we're going to put 10 times more. And if you're not changing the turnover number, or if you're not changing the relative affinity of the enzyme for its substrate, you're really not going to be affecting the reactivity that much, especially if it's an enzyme that has a high energetic shift to begin with. Skipping over some of this, I wanted to show you another thing about these the CTG clade. Unlike Saccharomyces cerevisiae, which does not natively use cellulose or cellobiose, in the CTG clade you find these pairs of endoglucanases paired either with beta-glucosidases or with or transporters for oligosaccharides, and they're just really just chock full. Everything you see there in purple are examples from the CTG clade. Everything else is missing. If you look here, this is the CTG clade, and these are the ones that have the cellulases. There is one exception, and that's Candida utilis, and when you actually go in and look at the where the single enzyme for Candida utilis came from, it turns out that it's borrowed from the CTG clade. The one exception here is Lipomyces starkii, which I said is down at the bottom. Another thing that's very interesting about Lipomyces starkii, and also with respect to Pichia stipidus, is they have they're just absolutely loaded with starch-degrading enzymes. And I'm going to shift some of the most lipogenic yeasts are closest to the base of divergence from the filamentous Ascomyces. Usually when you're talking about lipogenic organisms, you're actually dealing with Basidiomyces, Rhodotorula, and such. But the Lipomyces, and to a certain extent, Yaroia lipolytica, are truly lipogenic. One of the things, I won't go through all of this, but basically this is the pathway for lipogenesis. And it, starting from glycolysis, acetyl-CoA gets shunted in through citrate synthase, which then passes across from the mitochondria into the cytosol, gets split back, and then malic enzyme produces the NADPH that drives the fatty acid synthases. Now, fatty acid synthesis differs in bacteria, plants, fungi, and animals. In bacteria and plants, you have these 4 different subunits, and the carrier protein is a separate. But in fungi and animals, you have basically a single large fatty acid synthase, and the tasks are performed by one long protein that exists in the cells. What makes Lipomyces interesting, however, is that they actually have 3 complexes that look like fatty acid synthases. Now, we're not sure which of these is the most important. We think it's each of these are made up of an alpha and a beta subunit. But in the case of Lipomyces starkii, these are in a bidirectional operon, basically. And not only is it true for the fatty acid, every eukaryote has fatty acid synthases and a dimer, but usually they're scattered around through the genome. These are in one place, and also the ACL1, ACL beta, alpha beta subunits are also complex. The phylogenies of the ACL1 and ACL2 appear to be similar until you look at the distances, and you realize that ACL1 is actually very stable in whereas ACL2 appears to have a great deal of diversity, which might account for the differences in lipogenesis among these various organisms. There are numerous potential gene targets to increase lipid synthesis. I won't go into all of them. I'll simply say that we've developed a transformation system and complexities, and we can transform these cells, and basically this is one example of how we've doubled the production rate. This was a draw-and-fill fermenter run at a 3-liter level, and it was over in 48 hours, filled it back up, and hit it again in 96 hours. And some of these cells, you can see here, they're just chock full of liposomes. And the one thing that is a little bit disturbing is as these cells become lipogenic, they cease growing. So it's difficult to scale this process up, and that's part of the trick to everything is how do you do the processing. At this point, Xylome is working more toward its interest in synthesizing omega-3 fatty acids. Now, there are basically two different pathways that one can use in fatty acid synthesis. One is what we call the conventional pathway, and this has been used and engineered into Yaroia a number of years ago. Steve Piccottaggio, who incidentally was a classmate of both myself and my colleague Tom Kelleher. Steve Piccottaggio engineered this in Yaroia a number of years ago for DuPont. But then the more interesting one is this long-chain polyunsaturated fatty acid synthase pathway called PUFA-PKS. And it is found in a very small group of, I don't know what to call them, they're chytrids. Throstochitrid Throstochitrium is the one that's probably best known and best studied. But these enzymes are they're very large, and the biochemistry of them are not as well understood. In closing, I'd simply like to thank the many colleagues which I have gained through JGI, and if there's any time, I'll take questions.